Hello, welcome to the Robotics for All YouTube channel. Today's video is part of a series of videos covering introductory biology topics. Today's video concerns the cell membrane. It is important to note that prokaryotes do not have a cell membrane while eukaryotes do. Now let's discuss properties of the cell membrane. From our last video, we will note that the cell membrane surrounds the cell, semi-permeable, which means it controls movement of substances in and out of the cell, is made of lipids, proteins, and cholesterol, and is also called a plasma membrane. The cell membrane is also semi-permeable, which means that it controls what moves in and out of the cell and allows the cell to maintain a stable internal environment. Some terms to know is 1, hydrophobic, which means that stuff is insoluble in water or repels water, and is 2, hydrophilic, which means something is attracted to water or dissolves or shows an affinity to water. Osmembrane is a phospholipid bilayer, which means that the phospholipid, which means it is made out of lipids, or essentially fat, and it's a bilayer, which means it has 2 layers. It is composed of a hydrophilic head and hydrophobic tails. From this image, you can see that there are hydrophilic heads on the outside, hydrophobic tails on the inside, and then once again, hydrophilic heads in the interior of the cell, facing the interior of the cell. On the outside is a hydrophilic environment, on the inside is a hydrophobic cell membrane, and on the interior of the cell, or facing the interior of the cell, is a hydrophilic cell interior. Now let's discuss transportation. There are different types of transportation. There's passive transportation, which means that no energy is required, and is a natural process. There's facilitated transportation, which means that no energy is required and proteins help. And there's active transportation, which means energy is required and proteins help. First, let's discuss diffusion. Diffusion is the movement of molecules from areas of high concentration to areas of low concentration. It results in the even distribution of molecules. It is a form of passive transportation. Small molecules and nonpolar molecules use simple diffusion. Now let's discuss osmosis. Osmosis is the movement of water through a selectively permeable membrane from areas of high concentration of water to areas of low concentration. Many solutes dissolve particles like salt and sugar are not able to move through the selectively permeable membrane. The movement of water through the cell membrane can even out the concentration of solutes. There are different types of solutions. There's number 1, hypertonic solution, which is in which the concentration of solute particles is higher than the neighboring solution. Hyper equals above. And there's an isotonic solution, which means that the solution is a solution in which the solute concentration is equal to that of the neighboring solution. Iso means equal. And then there's a hypotonic solution, which means that the concentration of solute particles is lower than the neighboring solution. Hypo means below. If you want to, I'd suggest that you pause the video here and answer what kind of solution each of the following images are. Now the answers. The first one is an isotonic solution. The second one is a hypertonic solution, and the third one is a hypotonic solution. Now let's discuss facilitated diffusion. Facilitated diffusion is the diffusion of solutes through transport proteins in the cell membrane. Facilitated diffusion is a type of passive transport. Even though facilitated diffusion involves transport proteins, it is still passive transport because the solute is moving down the concentration gradient, as shown in the diagram here. Now let's discuss active transport. Active transport requires energy in the form of ATP. The energy is used to transport materials from areas of low concentration to areas of high concentration against the concentration gradient. Now let's discuss other modes of transportation. For one, there is the endomembrane system. The endomembrane system is the process of transporting proteins made by the cell outside of the cell. The protein is number 1, created at the ER, then the protein buds off the ER or endoplasmic reticulum in a transport vesicle. Then this vesicle arrives at the Golgi apparatus, undergoes a transformation, and is set to go outside the cell in another transport vesicle. The endomembrane system is the process of transporting cells made by the cell outside the cell. Number 1, the protein is created at the ER, the central dogma, and then the protein buds off the ER in a transport vesicle, and this vesicle arrives at the Golgi apparatus, undergoes transformations, and is set to go outside the cell in another transport vesicle. The endomembrane system uses endocytosis. This is used to take different substances into the cell, and exocytosis, which is used to take different substances out of the cell. Now let's discuss endocytosis. Endocytosis is another form of active transport to move substances that are too big for carrier proteins. The process of endocytosis uses transport vesicles that are formed from the cell membrane. These vesicles carry large substances such as proteins and sugars into the cell. Now let's discuss exocytosis. Exocytosis is the movement of large molecules out of the cell. Exo means out. Exocytosis releases substances from the cell by transport vesicles that fuse with the cell membrane. This is the last thing to note in our video. Thank you so much for watching this video on the cell membrane, and we hope you check out our other videos on introductory biology topics.